In my last video, we discussed the assembly and structure of DNA. Now we can discuss how DNA replicates. After all, one of life's major functions is reproduction. And reproduction starts with DNA's ability to make copies of itself. In cells, once the DNA has copied itself, the cell can divide into daughter cells, with each daughter cell getting identical copies of the parent cell's DNA. The whole process is called cell division. A cell goes through a cell cycle consisting of growth and reproduction. The cell cycle of eukaryotes can be divided up into 5 stages: G0, G1, S, G2, and M. Post-division rest is stage G0. Cell growth starts at stage G1 or gap 1. DNA replication takes place at the synthesis or S stage. There can be more cell growth in stage G2 or gap 2. The cell division stage is called the M stage or the mitosis stage. Both single-cell organisms and multicellular creatures have their own cell cycles. Single-celled organisms undergo cell division to increase the population. Multi-celled organisms undergo cell division to grow the organism or to replace worn or damaged parts of themselves. Asexual cell division is called mitosis. In organisms that reproduce sexually, the mitosis stage is replaced by meiosis, where each parent cell develops a haploid cell, a cell with only single-strand chromosomes. It is during meiosis and haploid cell formation that there is crossover, where sections of DNA leave their birth chromosome and swap places between the chromosomes donated by their own mother and father. A DNA mixing occurs, and recombination into a single-strand chromosome that is a mixture of both parents. In sexual reproduction, each partner donates a gamete, haploid cell, to form an offspring diploid cell that has a mixture of DNA from its 4 grandparents. That diploid cell will become the parent cell for a whole new population of single-celled organisms, or a completely new offspring organism that shares characteristics of both parents and its 4 grandparents. I shall talk more about meiosis, haploid cell formation, and DNA crossover in a later video. The DNA copying stage of the cell cycle is called the S stage or DNA synthesis stage. In DNA synthesis, the parent DNA double helix is split into 2 original strands. Each original strand is copied in a complementary fashion by adding complementary nucleotides to each original strand. A nucleotides are complemented by T nucleotides. G nucleotides are complemented by C nucleotides. The 2 new double strands are called daughter strands. So, each new double daughter strand of DNA becomes a mix of original and complementary new strands. The replication of DNA does not start in a single place. It starts in multiple replication sites. Initiation enzymes focus on regions where there are long sequences of AT pairs. If we remember, the GC pairs have 3 hydrogen bonds, but the AT pairs have only 2 hydrogen bonds. This makes opening up at DNA regions with lots of AT pairs require less energy to break those hydrogen bonds. Pre-replication enzymes seek out areas with high concentration of AT pairs, and where they pull open the parent DNA strand are called origins of replication. The replication sites are called replication bubbles, and there will be multiple replication bubbles on DNA that is replicating. The figure shows multiple replication bubbles on a DNA strand. The forks in the replication bubble open up more of the parent strand. The process of the replication forks opening up the replication bubbles is called elongation. The daughter strands are called leading if the copying direction is in the 5' to 3' direction. The daughter strands are called lagging strands if the copying direction is in the 3' to 5' direction, as shown in the figure. Assembly of daughter copies can only occur in the 5' to 3' direction. So further discussion of the assembly of leading and lagging strands is necessary. To begin the assembly process, the original nucleotides are read in the 3' carbon to 5' carbon direction. The new complementary nucleotides are added in the anti-parallel 5' to 3' orientation, so that each double daughter strand becomes an exact copy of its anti-parallel parent strand. The new nucleotides are always added in the 5' to 3' direction, so that the 5' PO4 is added to an existing 3' OH end as shown in the figure. The original parent DNA strand and the leading and lagging daughter strands are shown in the next figure. In this figure, there is a labeled drawing of an original parent DNA strand and the synthesis of leading and lagging daughter strands. First, the double helix of the parent strand is unwound and straightened by the topoisomerase enzyme. The topoisomerase can cut and rejoin the parent DNA as needed to uncoil and straighten the twists in the double helix. Next, the helicase enzyme uses a lot of ATP to break the hydrogen bonds linking the bases. The helicase enzymes split the parent strand into single strands. The binding proteins hold the single strands apart as the replication fork. The DNA polymerase enzymes on the daughter strands add the new nucleotides onto the single strands, A to T and G to C in complementary fashion. Replication of the leading strand is a straightforward operation. At the origin of replication, a primase enzyme adds a few complementary primer RNA nucleotides onto the exposed leading strand DNA nucleotides in the 5' to 3' direction. The primer RNA nucleotides will be removed and replaced by DNA nucleotides later during the proofreading step. A polymerase type 3 enzyme does the work of adding DNA nucleotides onto the sequence. The polymerase 3 enzyme needs a 3' OHN nucleotide to begin its work, and it uses the primer RNA nucleotides to begin. The polymerase 3 reads the single strand in the 3' to 5' carbon direction, and adds the complementary nucleotides in 5' PO4 to the 3' OH end of the previously added nucleotide. After the polymerase 3 adds many other DNA nucleotides to the leading strand, the polymerase 3 will go back and proofread its work. A's pair with T's, G's pair with C's. If the proofreading step discovers a mistake, say a new T paired to an original G, it will know there is a mistake. The polymerase 3 enzyme will come in, cut out the T mistake, and replace the T with a C nucleotide that is supposed to be paired with the original G nucleotide. The RNA primers also need to be switched out for DNA nucleotides. The removal and replacement of RNA primers is done by polymerase type 1. The leading polymerase 3 will continue to add DNA nucleotides in the 5' to 3' direction until it meets a lagging polymerase enzyme adding nucleotides in the 3' to 5' direction on the same single DNA strand. When leading polymerase meets a lagging polymerase, a DNA ligase enzyme will come in and catalyze a phosphodiester bond between the 5' PO4 of the lagging nucleotide to the 3' OH end of the leading nucleotide sequence. The lagging strand is a bit more complicated because it reads in the 3' to 5' direction. But the newly assembled strand ends in a 5' PO4, not a 3' OH. To get around this problem, the assembly must be done in short fragments of mixed RNA and DNA, called Okazaki fragments. Okazaki fragments are a messy solution. Nature is not always straightforward. Lagging strand replication has all the elements of leading strand replication. But because the lagging strand must be assembled in piecewise fashion, the lagging strand assembly goes more slowly. As the helicase does its work of breaking apart the DNA strand ahead of it, the lagging strand develops a long stretch of unpaired original nucleotides. At some point, a primase enzyme will come in and start to backfill the open stretch of unpaired nucleotides in the 5' to 3' direction with a short stretch of RNA nucleotides. This RNA will be cut out and replaced later. A DNA polymerase 3 will come in and add DNA nucleotides onto the primer RNA in the 5' to 3' direction. The DNA polymerase 3 will continue to fill in the unpaired stretch of the original lagging DNA strand until it reaches the section of daughter lagging DNA that is filled. At this point, it will stop. The whole mixed section of new DNA and RNA primers is called the Okazaki fragment. A cartoon of the Okazaki fragment on the lagging strand is shown in the figure. A DNA ligase will come in and form a phosphodiester bond between the new lagging strand and the Okazaki fragment. A polymerase III will come back and proofread the new segment to be sure it is correct. After that, a polymerase I will come in and cut out the RNA primers and replace them with DNA nucleotides. The helicase will continue opening up the original parent DNA in front of it until it meets another replication bubble and helicase working its way in the opposite direction. When the lagging strand meets the daughter leading strand coming from the opposite direction, a DNA ligase will come in and join the daughter leading and lagging sequences with a phosphodiester bond. A DNA polymerase I will replace the primer RNA with DNA nucleotides. The DNA polymerase I goes back, proofreads, and corrects any errors in the lagging strand. With both leading and lagging daughter strands proofread and corrected, we have two complete daughter copies of the original parent strand. We can now go into detail about termination. Termination occurs when two replication bubbles meet. At the meeting point, the 5' to 3' leading daughter strand meets the 3' to 5' lagging daughter strand. The helicase enzymes are finished and float away. Once the polymerase finishes adding the last DNA nucleotide, a gap still exists between the daughter strands. A ligase enzyme comes in bond. The daughter ends together with a phosphodiester bond between the 5' PO4 on the lagging strand end and the 3' OH on the leading strand end. Termination also occurs when the helicase enzymes reach the ends of the chromosome. The ends of a chromosome are called telomeres. When a DNA copy or daughter strand ends, there is no problem if the last new nucleotide ends in a 3' OH. There is a problem if the new nucleotide ends in 5' PO4. In the figure, the 4 scenarios of daughter ends are shown. The ends of the two daughter strands are missing nucleotides at the red backbone copies. In the top left, the daughter strand is missing one nucleotide. It ends in a 3' OH. There is no problem in adding a final nucleotide to complete the top left end. At bottom right, the scenario is the same. The daughter strand is missing one nucleotide. It ends in a 3' OH. There is no problem in adding a final nucleotide to complete the bottom right end. The top right end and bottom left ends have the problem of ending in a 5' PO4. The top right end is missing two nucleotides. There is no 3' OH to add to. The waiting red backboned nucleotide can't be added. The same scenario with the bottom left end. The bottom left end is missing two nucleotides. There is no 3' OH to add to. The waiting red backboned nucleotide can't be added. There is no straightforward solution to this problem. The DNA replication process just leaves off the last few nucleotides that can't be bonded to. One workaround to the problem of lost nucleotides at the DNA strand ends is to add lots of non-coding nucleotides to the telomeres of the original parent DNA chromosome. These non-coding DNA nucleotides on the telomeres can be sacrificed every time the cell divides. There is no problem if the DNA lost is not part of a gene that codes for an essential protein. Once DNA code for an essential gene is lost, the cell will cease to function properly. The cell may die or be forced to commit suicide by apoptosis. Apoptosis is a process by which a cell kills itself by a systemic release of enzymes that dissolve the cell. Telomere shortening is illustrated in the figure. The figure shows the ends or telomeres of a chromosome shorten every time the cell divides. The DNA is sacrificed at every cell division. Once the telomere shortens to a point where protein coding is affected, the cell will die. Cells that must divide frequently can reverse telomere shortening by adding nucleotides to their telomeres. Cells that must frequently divide include sex gametes, embryonic stem cells, and hepatic stem cells or blood stem cells. These cells activate an enzyme called the telomerase. The telomerase seeks out a DNA sequence on the 3' OH end of the telomere that is a frequently repeated sequence, TTAGGG. This telomere end sequence can be memorized by the mnemonic device, "Tell them all genes gotta go," or TTAGGG. When the telomerase enzyme finds the TTAGGG sequence at or near the 3' OH end of a telomere, the telomerase hydrogen bonds a complementary RNA sequence to it, AAUCCC, along with several extension RNA sequences of AAUCCC, AAUCCC, AAUCCC. The telomerase enzyme will then use the repeating AAUCCC sequences to assemble a complementary sequence of DNA onto the 3' OH end of the telomere. That complementary DNA sequence being TTAGGG, TTAGGG, TTAGGG. The process of forming a sequence of DNA by reading a sequence of RNA is called reverse transcription. A DNA polymerase can then fill the unfilled daughter nucleotides in the gap between the 5' end of the DNA daughter strand starting from the 3' OH end of the RNA sequence. That was put in place by the telomerase enzyme. Now this unfilled section of telomere is filled with DNA nucleotides, where it had been impossible to fill the unfilled section before the telomerase got involved. A ligase enzyme can come in and catalyze a phosphodiester bond between the previously existing DNA 5' PO4 end and the newly filled DNA sequence that used the telomerase at the RNA 3' OH end as a starting point. This process of extending the telomere could be repeated by relocating the telomerase further up the 3' OH end, a second or third time if a much longer telomere is needed. The action of telomerase is illustrated in the figure. The figure shows a telomerase adding RNA nucleotides to a telomere. In the final step, the DNA polymerase uses the RNA nucleotides to backfill the lagging strand with DNA nucleotides. Now, we said that cells die when their telomeres become so shortened that cells' chromosomes lose DNA sequences that code for functional genes. We showed that stem cells and other cells that must frequently divide can use telomerase to extend their telomeres. But most cells in the body don't have active telomerase to extend their telomeres. And over a long period of time, and through many cell divisions to maintain the function of whatever body part they belong to, these cells will shorten their telomeres to the point where they are encroaching on their functional gene sequences. Without the functional genes to make new proteins, the cells die. If enough cells die or cannot reproduce to new cells to maintain organ function, the organism itself will die. The shortening of telomeres is one aspect of aging, growing old, and the death of organisms, including people. Why can't we extend telomeres or activate telomerase in all cells to extend youth, or to extend the human lifespan? I assure you there are research scientists studying how to activate telomerase in all human cells, including scientists at the National Institute of Health (NIH) and at Stanford Medical Center. One roadblock to their progress with telomerase research is cancer. Cancer cells activate their own telomerase so they can endlessly divide and grow larger cancerous tumors. Cancer cells can continually divide and grow because they don't die a natural death like non-cancer cells. Until medical research gets better at controlling or eliminating cancer, there's a limit on trying to prevent cell death with telomerase. Once the cells have 2 copies of their chromosomes through DNA replication, the cell cycle continues through the G2 growth stage followed by mitosis and cell division, and the cell cycle repeats. That is the cycle of life.
And that is my take on DNA replication. In my next video, I will discuss sex, meiosis, and haploid gamete formation. Thank you for watching. If you enjoyed this video and want to help me make similar content, please like and subscribe.